Okay, guys, let's do it. So clearly it's an exam day from the attendance. Okay, so let's do this quickly, and then hopefully you guys can get out of here early and go and have a nice, luxurious lunch before you get tortured by me at 1 o'clock. There's no torture in this class. Okay, so exam freeze this week. For those of you who didn't know that, well, you're probably not here either, so that's fine. Oral exams are on Wednesday. I actually have a record number of people taking oral exams, so at least 5 or 6. I might have 1 spot left if anyone is still considering. Stump the teacher. We did a stump the teacher. I've graded a few of them. I didn't get through the rest of them. About kinesin 13. About how it's a catastrophe factor, but it's called kinesin, and is it motor protein or is it a catastrophe factor, or what's the difference? And so a lot of you guys found some wonderful things. This is one paper I found, which maybe some of you guys may have found also. And basically what it talks about is how the domain organization of kinesin 13 makes it so that it only interacts strongly with the ends of microtubules, and then that interaction creates a bending of those microtubules in such a way that it doesn't have strong contacts with the other alpha beta tubulin dimers. And so this is kind of just showing how that looks. Here's your microtubule, and here it's showing kinesin 13 binding, but actually up here it binds in such a way that creates that curve, and that's kind of what helps to disassemble the microtubules. And this is also kind of showing microtubules. This is electron microscopy. That's amazing, right? Seeing proteins. When you're talking about motor proteins in cytoskeletal structures, you actually can use EM to see them. Usually you can't use EM usually to see just like big proteins. Huh? How strong is that? Like, how strong? What do you mean by how strong? Like, what's the resolution?
Yeah.
I think it's 1 nanometer, I think they're saying. It's pretty phenomenal. But, and so here they're showing, like, these are your microtubules bound by the kinesin 13. So it's creating that kind of bending shape. And microtubules should not be bent like that, right? That's not a stable structure for microtubules. So those of you who sent that to me referenced will get a point. Okay, so let's take our 5 minutes to write. I specifically chose something that will benefit those who are coming to class on the day of an exam, because this is stuff that's going to be on the exam, and it gives you a chance to think about it and review it while we sit in class together. Okay? Compare and contrast the motor proteins that we discussed in class. Consider the kind of track they walk on and the direction they walk in. Which motor protein would you expect to be associated with vesicular transport? What about if you're going from the ER to the Golgi? What if it's retrograde transport? What motor proteins would you expect to be involved in mitotic spindle assembly or cell motility? So those are all different things to consider, and the way to approach it is to think about which cytoskeletal structures are involved in these processes. So go ahead and take a few minutes, and then we'll discuss. Okay, so that was 5 minutes. So Emily, actually now this is going to be the fun day where I get to decide who's here and who's not here. Monica, I saw you. There you are. Okay, so Monica, tell me, just tell me how you approach this and how you decided to answer it.
So it says comparison class, and I thought my motor proteins, so I thought about motor proteins. So I looked up what they were, and I found kinesin and dynein.
Kinesin, dynein, and what else?
Probably some more, I didn't know.
Okay,
And then,
myosin.
and what?
Myosin.
And my, yeah.
Yes. Okay, so you thought of those. Okay, so and then what did you do?
And then, I didn't really compare and contrast them, just, yeah, per se, I just listed them, and then I just like kind of skipped that, and then went to the other question and said, what kind of track did they walk along, and I said microtubules.
Okay, the ones you listed are microtubules and myosin is actin. Okay.
And I didn't get into it.
Okay, good. That's a good start. Okay, Savannah.
So I put that the dynein and the kinesin would be in microtubules and the myosin would be in the actin. And myosin 5 is associated with the vesicle and organelle transport, and it moves to the plus end. And the kinesin aids in the mitotic spindle assembly. And the dynein walks to the minus end, so that would probably be associated with the retrograde transport. And myosin 1 and 2 are also associated with cell motility.
Okay, good. Does anyone want to add? Yeah.
Doesn't kinesin, doesn't that move from the outside to the inside?
Kinesin is to the plus end.
Plus end that moves to the minus end?
Dynein walks to the minus end, and kinesin walks to the plus end. So kinesin walks to the plus end. The plus end is at the periphery of the cell, and the minus end is associated with the central one.
And so that when it goes to the plus end, that's using ATP?
They all use ATP. All of them use ATP. When you're moving to the plus end, you have to be, the only way to go to the plus end is to be associated with a motor protein that has the capability of walking to the plus end. And so that would be kinesin. Yeah. Yeah.
Would all three of them be involved with vesicular transport?
You could say all three of them are involved with vesicular transport. Yep. Yes.
Myosin also binds to the plasma membrane, and it's involved with cell surface perfusion, so also moving.
Myosin also binds to the plasma membrane.
Is that what you meant by cell motility?
Yeah.
Okay, sorry.
And is involved with cell protrusions and movement. Yep. Yep. What else?
Would you find these motor proteins within organelles?
Would you find
Or is it
motor proteins inside organelles? So I'm always
In the
scared of
Golgi-
saying no.
Because in the Golgi we have the cells. How are they?
Okay. So the question was, would you find motor proteins inside organelles? And we are going to cover this today. But I like to talk about these things. Or are they in the cytoplasm? And so you said they're in the Golgi, they're associated with the Golgi. And so your question is if you've got vesicles associated with the Golgi, how are they moving? So who wants to speculate? Well, you guys are all ready for an exam on this, so. Yes.
The first thing that came to my head was to know that we wouldn't find it on the organelles because the motor proteins are responsible for moving the organelles in the vesicles. So I feel like they would be associated, but they wouldn't be in the organelle.
Okay, so
That's the example of mine.
they would be associated but not in the organelles. Okay, and that's kind of what I'm thinking. I'm always apprehensive about saying definitively know anything because then there might be some motor protein that does something within the ER. I don't know, but usually remember you've got your motor domain that is walking along the track, and then you've got your tail that's associated with the cargo, and so it would be binding to your organelle or your vesicle on the outside, usually through a receptor, actually. Huh? Any other thoughts? Question? Yes. Which one is associated with the ER? So ER to Golgi, what do you think? What direction is that moving? So the plus end to the minus end, if you're going from the ER to the Golgi. So that's the plus end. What motor protein walks to the plus end?
I wrote
Kinase end. Yep. Retrograde is going in what direction? To the plus end or the minus end? Minus end. And what motor protein just walks to the minus end? Minus end. Right. Yes. Could you have said myosin for the ER? So myosin is actually involved in vesicular transport as well. I'm not 100% on the exact which myosin, but it would not be wrong. Yes.
So with microtubules, presbyopia is really straight up too much. Plus, minus is in the center, plus it is on the fourth one.
Right.
But with active,
Actin's different.
where it's like it's no concrete
Right.
place to say here's the minus and here's the plus.
Right. Right. So I wouldn't ask that question because it's not as easy to answer, or it's not as obvious and direct as an answer. So the question was, okay, well, with microtubules, you have a microtubule organizing complex, your centrosome, and so it's very clear where your minus and pluses are. Just think about that. Whereas actin is not necessarily in a specific direction, so how would you know? So I probably wouldn't ask a question that was so wishy-washy in the answer because there are different myosins, right? Most myosins walk to which end? Okay, but still, that doesn't really tell you much. Right. So that's why I wouldn't ask that kind of question. Yes.
For cell motility, would you mind because it is involved with cell evolution?
Huh?
Would cell motility, would that be myosin because myosin says that it is involved in cell migration?
Yeah, cell motility would be myosin because it's involved in cell migration. So you've got your front of your cell that's moving forward, and that's your arc, and your fulfillment, and your making those webs of actin moving forward. But the rear of the cell, you do have formin nucleated actin, and you do have myosin helping to contract and bring the rear of the cell forward. Yes. Okay. Are we good? How does everyone feel about the exam now? Good? Great. I just heard great, we're ready. Okay. See, I hear what I want to hear. Everyone's ready. Okay, so this is what you studied for the exam. I'm going to still keep adding to this because we're going to finish up the we're going to finish up the cytoskeleton over the next week or so before we move on to cell cycle. So the only thing that we did the other day was those proteins that organize cytoskeletal filaments, so all those active cross-linking proteins, and proteins that anchor the cytoskeleton to the periphery or to different parts of the cell. And then we spoke a little bit about motor proteins. And so I wanted to give you guys this link for different motor protein movies. This is a website from a lab that works on motor proteins, and there are a lot of helpful resources. I know you're like, great, thanks, taking the exam today, we don't care. But, you know, not everyone's taking the exam today, and then there will be more about motor proteins in the final. So it will be useful. I'm not going to go through them all now. It's a resource, the link is here. Okay, so we spoke about 3 different kinds of motor proteins, kinesin, dynein, and myosin. And so I wanted to just do like generally similar differences between them, right? So what's similar among all 3 is that there's some sort of nucleotide hydrolysis that needs to happen. So that's ATP hydrolysis, and that ATP binding, ATP to ADP, the release of that phosphate, all of that is associated with conformational changes that change the structure of the protein and result in movement. That's how they work. What's different is the conformation of the proteins in the different nucleotide bound states differs a little bit among the different motor proteins. But just understanding it's that nucleotide binding hydrolysis release process that's associated with movement is important. The other thing that's similar among all 3 is that those conformational changes, they're small, right? ATP and ADP, they're different in size, and so there's this small change that happens, but it's amplified by different domains within the motor protein, such as the lever arm in myosin, the linker in kinesin, and the stop in dynein. Okay, you might be saying, what? And so there's this example in the book with myosin, right? So remember, there are many different myosins, and they have different structures. They have conserved head domain, but often that lever arm is different, right? The size is different. And that's going to result in a change in size of the step that they take. Does that make sense? Long legs, walk fast. Short legs, right? You know when you're walking with your 2-year-old and you're like, right? Who has a 2-year-old? There you go, you know. Represent, okay. So myosin 2 has this really short lever arm, so the swing is going to be shorter, so the steps are smaller. Right, myosin 2, what's that associated with? What does myosin 2 do? So motility, but what about, what else have we seen it with a little bit and you learned about in physiology? Muscle contraction, right? So that's the myosin. That functions as part of, that functions as part of the thick filament, and so you need a little small step, right? Because they're all arranged together, and it needs to take a small step and let go with myosin, right? Because you've got that bipolar thick filament, and all those heads are right next to each other. And so if they're moving really far, they're going to bump into each other. So you need to take a little bit of a step and get out of the way, because there's another myosin head nearby. Whereas with myosin 5 is more involved with vesicular transport. It acts by itself. And so it takes longer steps, and it spends more time bound to the actin filament. And so that difference in size is related to its function. Okay, so I told you motor proteins are involved in transport of membrane-bound organelles. Yes?
Do we need to know, like, when ADP or ATP, like, if it's tightly bound or loosely bound within each one?
Now, I mean, I just got the question is, do we need to know when, based on nucleotide binding state, exactly what is happening? No. Just understand that you have those changes based on nucleotide binding or hydrolysis and how that affects, generally, the conformation of the protein. And that's going to be amplified by the lever arm or the linker or the stalk or whatever. And so that's what's allowing for that movement. Okay, so movement of organelles. Okay, so here these are pictures coming out of the textbook. So if you don't understand it, go back, read the figure legend. Actually, I just want to take a minute to let you know that there was a surprising number of students who visited me for help in the last 2 weeks who were confused about things. And I said, well, did you read about it in the textbook? And they all said no. So I am happy to explain stuff to you, but please try to figure it out by looking at the textbook first, because teaching yourself is huge, right? But anyways, I'm always happy to help you. Textbook, expensive, right? You should use it. So, this is right out of the textbook, and here you've got two different cells, or one type of cell, either treated with nicotazole, which is a drug that depolymerizes microtubules, or not. So these are your untreated cells. And what we're looking at here are our microtubules, so those are red, and our Golgi, which is green. Okay? And so what's the difference here? What do you see happening here when you treat with nicotazole? Yep.
The Golgi isn't localizing where it should be. It's not against the nucleus, not been treated with compounds.
The Golgi is not localizing where it should be. It's not near the nucleus, right? What else? Does anyone else have any other observations? Huh? Three. They're just two pictures. One picture is cells not treated with nicotazole, and one picture is cells treated with nicotazole. They're not the same cells. They're two different dishes. One not treated and one treated, and they just selected a cluster of cells to look at. Yeah. Yes.
The little shape of the cell. Like the way the active.
Well, these are microtubules, the red.
I'm sorry, microtubules.
Okay, so the microtubules look different in these two. How do they look different? Yeah.
Here we have the, I guess, fibrous one, and here it looks like you just have individual, I guess, clumps.
Yep.
As opposed to
Looks like subunits. So here you've got actual, if they're lines, right? Those are your polymers. Those are your actual microtubules. Whereas here, it looks different. I just heard it looks like clumps. Maybe it's just subunits. Yeah, well, they're depolymerized. So yeah, it's all just subunits. And that's why you have this diffuse staining. So this tells you two things. One is nicotazole is working because you've got depolymerized microtubules, right? And then the other thing is that those microtubules are required for keeping the Golgi where it should be, Yeah. And how is the Golgi being kept in that spot right there? What's attaching it to the microtubule? Someone raise your hand. Yeah.
what was it? Transport, like actin or something. These are microtubules, right?
Microtubules.
So probably dynein.
Probably dynein. Why did you choose dynein over kinesin?
Because dynein is more towards the minus end, closer to the, I guess, where you find the central of the central.
So exactly, because dynein is closer to the minus end where your centrosome would be, so that's closer. That's a logical assumption. I'm not 100% sure if it's not kind of like a little bit between the two of them, but that's a very logical assumption. It would be, if it's to the minus end, you would probably have a motor protein that's kind of can walk in that direction to keep it there. Yes. Yep. So the question was, how come plectin, which is a cross-linking protein associated with cross-linking intermediate filaments and cross-linking intermediate filaments to actin or microtubules, how come that protein is not holding the Golgi near the nucleus? Well, what do you think? Well, so in order for a protein to do something, it needs to be able to bind to the thing. So chances are, well, first of all, it's the microtubules that are required for keeping the Golgi here. Why isn't it? So really, I'm going to change your question. Not plectin, not intermediate filament, but why isn't it one of these proteins associated with organizing the cytoskeleton, cross-linking protein, or something static? Why is it a motor protein? I don't know, but my guess is that at least why plectin is not involved is plectin has domains for interacting with intermediate filaments and domains for interacting with actin or microtubules, but probably doesn't have any way to interact with the Golgi. And so we're going to talk in just a minute at how these motor proteins interact with organelles or vesicles. Okay. Any other questions? Okay. So this is just a picture showing microtubules again, and just this concept of movement of organelles along the microtubule highway. Right, so you've got your minus ends at the center of the cell and your plus ends at the plasma membrane. And dynein moves towards the minus end, so anything moving inwards will be being moved by dynein. And kinesins move towards the plus end, so things that are moving outward will be moved by kinesin. And so here I have a question that's similar to what we just did for the writing exercise. What motor protein would be involved in transport of a vesicle from the Golgi to the plasma membrane? Someone raise your hand. I know you all know it. I hear before screen. I've seen you today. Who have I seen today? Yes. Golgi to the plasma membrane along microtubules. Kinesin, right? Because it's moving to the plasma. Good. So how do motor proteins transport cargo? We told you there's a head and there's a tail. Head walks on the track, tail binds the cargo. So this is kind of how they look. Here's your microtubule. Here's your different kinesins. And they have some sort of receptor and protein linker, or just receptor. Usually it's a receptor associated with the cargo, like so it would be a vesicle or membrane-bound organelle cargo, right? So you've got a receptor there. Where's that receptor made? In the ER, right? Oh, connected. Okay. And then that receptor, the cytoplasmic side, is going to interact with your motor protein, or sometimes there's a linker protein, but either way, somehow it's going to connect, and then it can walk along microtubules. Same with dynein. Here you also have receptor, some kind of protein complex linking to the motor protein that can walk along the microtubules, and then here you see myosin, same thing, okay? So kinesin, motor protein tails, and light chains attached to cargo through membrane-associated motor receptors. That's what they're called. These receptors are called membrane-associated motor receptors because they bind to motor. The cargo needs to go to different places. How is this determined? Yeah.
Targeting proteins like RAD or
Right. So remember, you've got your everything we learned when we talked about vesicular transport applies here, right? Now we're just learning how those vesicles actually move, but you've got your RABs and you've got your snares that are involved with diffusion. Yep. Where would you expect to find motor receptors? We just kind of mentioned this. Where would you find them in the cell? If you stain your cell with fluorescent green antibody against a motor receptor, where would you find them? In the ER, because that's where it's being made, and where else?
It's on the cytoskeleton.
On the on motor receptor? Oh, on the cytoskeleton, okay, because it's going to be moving along. Okay, where else? Golgi. Basically, all those membrane that's being made in the ER, that membrane is going to become the Golgi, just what we've learned about, right? So that's where they should be. So there are these J and K interacting proteins, and they are scaffold proteins involved in cell signaling, and they function as kinesin receptors. So what cellular process do you think that they might take part in? Export kinesin plus N. So some sort of export, some sort of signaling function. Okay, so here this is showing you exactly how it looks. So you've got dynin right here, walking along the microtubule, and here it's associated, here's your membrane-associated motor receptor, this green one here. Then there's this huge protein complex. Here it's the dynactin complex that is interacting, basically linking your vesicle to your dynin. And so actually, apparently, not just vesicles, also membrane-bound organelles, but organelles tend to be coated with these proteins. So they're kind of sitting there available for interaction with other proteins. Yes?
Wait, so the dynactin complex is a receptor?
The receptor is the green part.
Oh.
The dynactin complex is actually a linker between the receptor and your motor protein. Any other questions? Okay, so myosin 5 is associated with vesicular transport. And so this example is melanosomes. And these what are melanosomes? Does anyone know? Pigment, right? Responsible for pigment. So it contains the pigment, right? And so melanocytes make melanin, and then they're transported in melanosomes to the end of out of the melanocyte, and then they can be delivered to neighboring keratinocytes that give the cell their color, right? So myosin 5 is involved with transport of the melanosomes to the dendritic processes of the melanocytes. So basically these kind of parts that are sticking out. And then they can be exported and delivered to neighboring keratinocytes. So what if you had a mutation in your myosin 5 gene? What kind of phenotype would you expect to see? No pigment or reduced pigment, right? So what domain of myosin 5 would have to be mutated for you to see no color? Cargo binding.
It could be both, even moving it.
Yeah, it could be both, right? Because I guess if the motor domain, the heads couldn't bind to actin, then it still couldn't travel. It might be able to bind to the cargo, but won't be able to travel. Or what if it's the ATPase, the ATP binding domain? Right. So it's important when you're thinking about mutation, think about where in a protein is something mutated and what effect is it going to have on the function of the protein. Okay, so another thing myosin 5 does is involved with mRNA transport. So this is an example of the role of cytoskeletal proteins and motor proteins in mRNA localization. So in this example, this is looking at a yeast mRNA called ASH1 that needs to be expressed in the daughter cell. And this is yeast, and this is budding yeast. So budding yeast, the way it divides is it forms a little bud that gets bigger and bigger and bigger, and then eventually it buds off, and then you have another yeast cell. And so this mRNA needs to be expressed only in the daughter cell. So the mRNA needs to be localized to the growing bud. And so what this is showing here is here's your RNA. And then it has some kind of structure in it that binds to a protein that's going to link to myosin 5. And so you have a linker interaction between the RNA and the motor protein, which can then walk to the plus end of this form-nucleated actin. And then that RNA is going to be over here, and this is where the bud's going to form. And so if you do immunofluorescence, not immunofluorescence, FISH, which is like immunofluorescence, but for RNA, where you use a probe that's specific to the RNA sequence you're looking at, you can see the ASH1 mRNA is localized to the bud tip. So here's your original cell, and here's your bud, and this is going to be your daughter cell here. And so up here is where that red is, because this is the same picture, just one of them is only looking at fluorescence, and this one is looking at the whole cell. Any questions about that? In situ hybridization. So basically that means inside the cell you're hybridizing a probe that's specific to that RNA, and it's fluorescently labeled. So it's a way to track and follow where certain RNA, but you can do it for DNA, you can do it for anything, but it's that interaction between the probe, which base pairs, with the RNA target. Okay, so just like everything in the cell, something needs to be regulated. It all needs to be regulated, right? So here's an example of like how myosin activity is regulated when it comes to bipolar filament assembly. So it's regulated through phosphorylation. So you'll have an inactive state, and so here your myosin is folded up, and it cannot assemble into a thick filament to participate in contraction. This is myosin 2 we're talking about. This is myosin 2 not in muscle cells here. Well, I guess it's regulated a little bit differently in muscle cells. This is when we're talking about myosin 2 assembly to form a contractile ring in mitosis, for example. And so here, when you get phosphorylation by a kinase called MLCK, myosin light chain kinase, you get phosphorylation of the myosin light chains, and that changes the conformation of myosin. So you get this extended tail, and that allows for spontaneous self-assembly into the bipolar filament, so you can get the contractile ring to be active. Okay. When would you expect when would you expect this to be active? When in cell cycle? Cytokinesis, somewhere in mitosis, right? Do you need a contractile ring and interphase? No. Okay, good. Okay. Here's some more regulation examples. So in fish, melanocytes can alter the location of their pigment granules in response to neuronal or hormonal stimulation. They could either be dispersed and look like this, or aggregated, so basically all in the center of the cell. So this changes changes the color of your fish, right? Changes the pigment of the fish. So this is regulated by phosphorylation of the kinesin light chain. So phosphorylation of the motor protein in response to a signal. So signaling results in phosphorylation of the motor protein. And so this inactivates kinesin and allows dynin to take over. So which direction does kinesin walk in? Plus end. So here, if you inactivate kinesin and allow dynin to take over, that makes sense, right? It's going to walk to the minus end, so it's going to aggregate the pigment granule. Questions? Who's ready for Thanksgiving? Who has been going at $5 Friday and getting they've got this Thanksgiving in a bowl thing going on? Oh, who knows about $5 Friday? Good. A lot of students do not know about $5 Friday, and I find it to be a tragedy. All of the dining halls, it's $5 all you can eat on Friday. That's what I'm saying. Oh, I know, you guys are going to like come here and graduate and go and never enjoy $5 Friday. So this is every single Friday. The actual dining halls, not the ones where you pay a la carte, but where it's, you pay when you walk in the residence hall. It's blend. Anyways, they've been doing this like Thanksgiving in a bowl. Only for lunch. $5 Friday for lunch. Yeah, so you guys are here probably hungry. Okay. Anywho, see you all on Friday. Phosphorylation of the kinesin light chain inactivates kinesin, and that allows dynin. So you can imagine kinesin and dynin are both available and associating with the microtubules. So if you inactivate one, that will allow the other one to take over. Okay. Oh, muscle contraction. So I'm going to very much go over this quickly because every year I feel like you guys have learned it so well in physiology, and then a lot of people are taking advanced phys and animal phys that we're not going to, not everyone actually, some of the wonderful MB3 majors get the shaft. So I don't go too crazy on it, but we're just going to go straight to molecular. Okay. So that's muscle. Oh yeah, right. So you've got a muscle that's made of muscle cells that are made of many, many of these myofibrils, and then each of them contains these sarcomeres. Okay, made up of sarcomeres. So that's kind of, we're going right there. Skeletal muscle cells are formed by the fusion of multiple myoblasts, and so they contain many nuclei. And so if you stain the nuclei with DAPI, so that this is a rat muscle, and you can see that there's many, many nuclei because they're made from the fusion of multiple myoblasts, which are individual cells. Myofibrils are made up of repeating contractile units called sarcomeres. And so this is how they're organized. It's pretty much just the organization of your actin and your myosin. Okay, and so your dark band here, in your sarcomere, that is your thick filament, so that's your myosin. And so this light band here is your actin. And so what's going to happen is you're going to get that movement. Everyone do the myosin dance. Yeah, you're going to get that walking along the actin at either end to the opposite directions, and that kind of brings in your, not the actin, that brings in your actin. And that's muscle contraction. So let's go a little bit more in, a little bit closer. So here's your sarcomere. You've got your Z disc, and then you've got your cap Z. Remember we learned about cap Z, capping protein, right? So here's your actin. So your actin is regulated in length because it's capped at one end by your cap Z and the other end with tropomodulin. And so then you've got your, and this is your plus end of your actin filament, and this is your minus end. And so then you've got your myosin that's assembled into your bipolar thick filament. Why is it called bipolar? Because why? Well, because it's gonna, the myosin, it's oriented in the opposite direction. So you've got one myosin where your motor domains are on this end, and then you've got your tail this way, and another cluster of myosins. So each little head is here, right? Another cluster of myosins with your head domains in this way, where your tail is oriented this way, and it's that assembly of your tails that create that bipolar thick filament. And your heads are going to walk to the plus end, so they're going to walk towards your Z disc, right? Does everyone agree with me? Okay. So your cap Z and triple modulins anchor and stabilize actin filaments. Then there's nebulin. Nebulin nebulin. So nebulin is a protein, a huge protein actually, that interacts with and determines the actin filament length, because you don't want willy-nilly actin filaments, right? Titin is a molecular spring that is going to be positioning your thick filament. So that's the spring-looking yellow one here. It's positioning your thick filaments midway between your two discs. So it all has to be lined up properly so you can contract. So that's a number of accessory proteins there that we've learned about. Okay, so muscle contraction is activated by release of calcium from the sarcoplasmic reticulum. So that's kind of this mesh coating around your myofibrils. And that calcium release is released from a motor nerve. So there's a signal from a motor nerve, you get release of calcium, and then what happens? So so you get this calcium release. What is it doing to initiate the contraction? I want to see what you guys know. Okay, I heard something about troponin. Okay, so someone knows something. Yeah. Right, so basically what's happening is you are moving your tropomyosin and allowing myosin to bind to actin. So this is exactly what's going on. So you've got your actin, so we'll keep zooming in, right? So you've got your actin, and then you've got your tropomyosin. And it's held in place by this troponin complex, which has 3 subunits, an inhibitory subunit, a calcium binding subunit, and a tropomyosin binding subunit. And it's all regulated by calcium. So as soon as calcium is released from sarcoplasmic reticulum, it can bind to the calcium binding domain, and that is going to basically release tropomyosin from exactly where it is on the actin, and it's going to slide over and expose the binding sites for myosin. And that allows contraction to happen. So when you're resting, this the troponin IT holds tropomyosin in the way of the myosin Q head, so it's blocking myosin binding. Then you get this influx of calcium. Calcium is bound by troponin C. Troponin I is released, and then the tropomyosin can slide out of the way, and it's available. You have your actin available for myosin binding. So this is kind of showing if you're looking down, or if I guess a cross section of your actin. So you've got your actin, and your tropomyosin is going to be blocking the myosin binding site. And when you add calcium, the troponin IT, that complex is going to allow tropomyosin to move out of the way, and the myosin binding sites are exposed on the outside. Any questions about this? In my experience, that question on the exam is almost universally correct for everyone because everyone's learned it so many times. So that's always nice. For those of you who have not learned it, come see me if it's confusing at all. Okay. So again, this is another way to look at it. I like this one because it reminds me of olives, and I love olives. Right? Don't they all look like olives? So this is your actin. And so normally your so all those, if you imagine those little holes being the myosin binding site, the tropomyosin is normally covering it, so it's preventing myosin from interacting. And it's being held there by this troponin complex. And as soon as the calcium comes in, it binds, and then you get movement of that tropomyosin out of the way, allowing for myosin to bind. Done.
Yeah.
Yeah? Is that really it?
If the last slide is,
Yeah, that's it. Okay, guys.